It's our very own Louis Kuhn, Dr. Louis Kuhn, who actually did his PhD, developed a probabilistic framework for the analysis of macromolecule organization in cryo-electron tomograms at the Max Planck Institute for Biochemistry and the Technical University of Munich for his PhD. And then worked afterwards as a data scientist at Roche before then joining Cubic, I think about 2 years ago when I'm right, where he's now working. And also, as you already heard earlier, he's working on imaging analysis and deep learning. So, and he will talk about biomedical image segmentation using modern machine learning methods. So I'm really looking forward to the talk, Louis, and the stage is yours.
Thank you very much for the nice introduction. Yes, so I am going to share now my screen. Hope that is correct. Can you see my screen?
Yes.
Okay, great. Then I can make it. Yes, so maybe take this away. So as mentioned, I will tell you a little bit about our efforts to develop machine learning methodologies for semantic segmentation of biomedical imaging. Specifically, we'll tell you about some use cases in cancer research. Yes, so as we started to integrate imaging support into our data management infrastructure into QPortal, we've also begun to work on hooking up the system to our workflow mechanisms. We want to be able to run workflows to analyze data, not only on omics data sets, but also on the imaging data. So we've been working on building reproducible workflows based on machine learning to analyze biomedical imaging data. And of course, we want these workflows to be reproducible. That is a big part of what we're aiming at. So we need to make sure the analysis of these methodologies is thorough. So first of all, as mentioned, it's important to develop methods that are that can be reproduced. So aside from developing these pipelines, these methodologies into reproducible workflows, we also need to make sure that we comply with best practices in machine learning. So that mainly involves, not only providing quantitative results, but also the data set that was used to train the methods, the trained methods themselves, the code, and everything that is needed to reproduce the work. So that is part of best practices. But aside from having reproducible best practices in the development of our pipelines, we also want to make sure that the algorithms that we deploy to analyze biomedical data are reliable. And what does that mean? So not only do we want these methods to be very high performing, we want them to be quantitatively state-of-the-art methods that can deliver very good results that enable research. But we also want these predictive models to provide more information about how they make the predictions that they do. In particularly, we want to make sure that a prediction from a machine learning model can be ranked or can be well, the uncertainty of its prediction can be quantifiable. That means that not only do we get a prediction, but we get a certainty of how the model is certain of a particular prediction, and that's quite important to decision making. Aside from that, we will also like to we would like these models to be transparent, so that we can understand why the model made a prediction based on the data of an input. So keeping that in mind, that we want to develop reproducible and reliable machine learning algorithms, we focused on one specific task to start building up our capabilities for running this analysis. Mainly, we focused on deep learning methods for semantic segmentation, and I will tell you about about deep learning methods, why we chose this, and I will tell you about why semantic segmentation is a very attractive um, problem, very attractive task that is widely needed in many biomedical imaging modalities. So why machine learning? Why do we want to use these so-called deep learning models to analyze imaging data? Well, there's a very compelling quantitative argument to be made, and this is usually a plot that is widely used. Here we can see that the performance of the best algorithms in the ImageNet competition. This is a very large competition, widely known, where computer scientists compete on developing methods to classify a big database of images. So there's a lot of images, and these images come with they're hand annotated, and the task is to classify a certain image according to the objects that it contains. And so as you can see, the error rate of these algorithms went from 28 to 25 in 2011 to 16 in 2012. So before 2012, mainly what was being used was traditional algorithms for classification. And after 2012, with the so-called AlexNet, deep learning algorithms were employed, and the performance dropped significantly. So the visual recognition capabilities of these deep convolutional neural networks became was exemplified in this challenge. And as you can see, over the years, with more iterations, as these models as these deep convolutional models started to add more layers, then you would see the performance increase and the error rate drop to the point that the error rate for some of these classification architectures is now below what is considered human performance, what has been measured. So this is a very compelling, attractive argument to use these techniques to analyze biomedical data. And there's also a qualitative argument. As you can see, there's also generative models that have been used to create synthetic images for video, which are now very hard to distinguish from real video. So it's so these visual recognition techniques based on neural network models have come to have very high performance in a variety of tasks. That's what makes them quite attractive. So and why do we want to use, why do we want to focus on semantic segmentation using deep learning? Well, that's because semantic segmentation is indeed a challenging task. It is more challenging than detection and localization itself. However, it provides a lot more information. So what is semantic segmentation? It's really classification of an image at the pixel level. This is a task that is needed across many microscopy modalities and medical imaging modalities. It allows you to detect biological structures and quantify their morphology. So it's pretty useful because it allows you to capture quantitatively the shape of objects. And it provides the means of doing high resolution spatial statistics. So that is quite appealing as well. And well, automatic segmentation, multi-class segmentation has many attractive qualities. First of all, that it is not influenced by human factors, and reduces segmentation time. Anyone who has had the need to segment images manually knows that this is very time-consuming and painstaking work. So this is alleviated, and of course, it can provide a second opinion for spatial annotations. Then how do we want to do it? How do we want to do semantic segmentation for a variety of medical imaging modalities? Well, there is a very successful model called the UNIT, which I'm showing here. This is how the deep neural network architecture of this unit looks like. It has been very successfully applied in many microscopy domains and medical imaging as well. It is quite general. It allows you to input segment two-dimensional and three-dimensional data as with an arbitrary number of channels, and it can provide multi-class segmentation map. That is that you're not restricted to the number of classes that you can segment. You can segment an arbitrary number of classes. So that's a pretty general. So as you can see here, mostly what happens is, well, this unit architecture is composed of convolutional neural networks, and as an image goes through a convolutional layer, it is abstracted into so-called feature maps. And so as you can see here, we would have an input of an image, and it would be it would go through these convolutional layers. The number of channels would grow as the feature map goes down into this network. The spatial resolution of the feature map is reduced, is halved, and the number of channels is increased. So there's computations that happen through these convolutions. You have this downsampling path which detects features, and this upsampling path where the opposite calculations happened. And at the end, you get an output segmentation map, which is a map that classifies every pixel in your image. And here is a small example of how binary classification would look like, the output. And here you can, as you can see, you can detect cells and also measure their shape and spatial organization with this information. So what do we want to do? Well, in order to apply this technique in a broader sense for many modalities, we decided to go for a semantic segmentation reliability benchmark. And the idea here is to benchmark the performance of a simplified version of the unit architecture. So as you can see, this simplified version is much more compact, much smaller. And the idea is to benchmark it for a variety of data sets. Importantly, we want these data sets to be public and published. We already heard about the importance of public data sets. So this goes as well into reproducibility. We want to make sure that this analysis is reproducible. So we want to focus on these data sets. The idea is to train a basic unit and then evaluate its performance against variants of the same architecture. So make more sophisticated versions of it and see if the performance, there's a significant performance increase, and also measure other properties of the network. as I mentioned, one of the things that we want to measure, not only the performance, is whether we can extract a measure of uncertainty of the predictions that we have. This is, as I said, an important property of a predictive model, to be able to quantify how certain a prediction is. And also we want to see if we can query the model and ask what are the features that are important for a particular prediction. And that goes into interpretability. On what information is the model making predictions? So these are all aspects of properties of a model that need to be addressed before we can roll out standard imaging analysis on medical data. So these are quite challenging analysis. It needs to be very well documented. So I will tell you about two basic applications of this analysis. One is for two-dimensional data, H&E stained tissue images, and there the task is segment nuclei. So it's a binary classification problem. And I will also tell you about the segmentation of liver and tumor in X-ray computer tomography. So yeah, these are both public data sets which have been made public and published in order to be able to benchmark many detection algorithms. So it is a good test bed to analyze these models on. So here are some results on the first data set. As I mentioned, H&E stained tissue images. And so the task here is to segment nuclei from very diverse set of histology images. And the idea here of this data set is to be able to train a classifier which can segment nuclei for tissue images of many organs, many disease states, and different. Also, be able to generalize to be as general as broadly as possible. Yeah, this is, as I mentioned, a published data set, and the highest performance is here marked. So here we applied a trained unit for as I mentioned, binary classification. The input was these 2D images with 3 channels, red, green, and blue, and the output was a two-dimensional mask. Here you can see qualitative results. Input, the label provided by the dataset, and the prediction of the model. Yeah, so this plot just shows you the performance of the model during the training process. As we train our model, here on the x-axis we have the epoch. So an epoch is one training cycle over the complete training data set. So as we train our model, we can see the performance of our model on the different classes. This score is so-called Jacquard index, the intersection over the union, and it's really a measure of performance of for semantic segmentation results. As I mentioned, it's quite widely used, and what it computes, it can be expressed this way, but it mainly computes the intersection over the union of the predicted mask and the reference mask. So the intersection over union over the label and the prediction. And as you can see, we are close the simplified unit. It gets a performance, stabilizes in the performance that is comparable to the state of the art, even though it's a very simplified model. And for, well, the background class is pretty high, but yeah, it provides reasonable results. We did the same thing for abdominal X-ray CTs, and there the idea was, there the task was to do three-dimensional segmentation of both liver and tumor. And this is quite important, in particular for tumor burden calculation. Again, this is an open task. This is an open problem. Segmentation, automatic segmentation, it's not easy, since there's different CT scanners and acquisition protocols, intensity changes, varying contrast levels, and there's many variables that make automatic segmentation a very challenging task for this modality. And so we approach the problem in the same way. We used a unit for a multi-class segmentation as a multi-class segmentation model. There we have three classes: the background, the liver, and the tumor. The input is a three-dimensional image with one channel, which is the intensity of the image, and the output is a three-dimensional classification mask. So here as well, we quantify the performance of the method during the training process. And as you can see here, the most important curves really are the test curves that tell you the performance of the model on the testing data set. So as you can see, for the liver class, we get a performance of around 0.9, which is pretty well. That is more or less what it's reported as the state of the art. And for tumor detection, it's a bit lower than what it has been reported. However, it's direct quantitative comparison. It's a bit difficult, since the model that was used to measure this lastly used 2D tomogram slices instead of a full three-dimensional convolutional neural network. So yeah, to give you an idea of how the results look like, here you can see a three-dimensional rendering of a tomogram from the testing set. Here, the liver class, the label of the liver class is rendered as a yellow mesh, and the prediction of the unit is rendered in purple. The same for the tumor class. You can see the red mesh for the ground truth, and the prediction of the model in purple. This is it's a bit cumbersome to visualize these results in three dimensions, so sometimes it's useful to use cross sections to analyze it, to take a look at the results. And here you can see the label, the contour of the label, the ground truth in yellow, and the prediction in purple. The same for the tumor class. So it seems like the network is not as specific. It lacks some specificity, but more or less it's able to detect tumors. So it's great that we are able to have these very neat results. But we would also like to not only get a prediction on our pixel class, but we also would like to get an uncertainty value in order to augment this information, to be able to say with statistical grounds how certain this prediction is. That's quite important for a predictor, especially in clinical settings. So this prediction uncertainty can be used in many ways, and it can be useful in different scenarios. For example, decision making, and making sure that if there's a prediction with high uncertainty, that this prediction is passed to a human. for verification. Uncertainty has also been shown to be quite useful for biological discovery, for aiming biological hypotheses. So particularly a class of probabilistic models called Gaussian processes, which are able to quantify uncertainty very naturally. They are also interesting because they are capable of adapting to new data. So you can always retrain your predictor when you have more information, and the prediction should be reasonable. So these type of Gaussian processes are interesting, and they've been shown to be able to guide biological discovery. And interestingly, so since these predictive models do not output a single prediction, but rather a distribution, you can use dispersion-related statistic, in this case, a standard deviation, to assess the uncertainty of a certain prediction. And so why are these models?
Just quickly, just if you could just try to come to an end, because we're over time already. Thanks.
Oh, we're over time. Oh, apologies. Okay. Yeah. So I will tell you very shortly about this, well, it's there's a way of approximating these deep neural networks to be able to provide a prediction provide an uncertainty value on your prediction, and this pretty much involves Monte Carlo dropout procedure, which is exemplified here. But for the sake of brevity, I can show you some results. We are indeed able to apply this methodology and get a reasonable uncertainty value for predictions. We can do that also for our three-dimensional unit, and we are able to get reasonable results. So for certain regions which are false predictions, we do see high uncertainty levels. So results look reasonable. And as I mentioned, we also would like to look into interpretability. How can we make this deep neural networks interpretable, mainly to highlight which are what is the importance of certain features for a prediction. So I will probably skip a bit fast over the slides. We use a technique called guided GradCam, which can give high resolution features and be very class discriminative. So it allows you to highlight what are the important features from your input that were able to that led to a specific classification. So as you can see here, we can apply this to nuclear segmentation, and indeed the features seem to come from indeed cells within nuclei. So this makes sense, and we also have been able to apply that for our 3D case, and indeed we do see that the important features for classifying pixels, detecting tumors and liver, do come from the objects in questions, and the network is not exploiting artifacts in the image to make predictions. So to come to a fast conclusion, I showed you that indeed a simplified version of the unit is able to get comparable results to the state of the art. That it is possible to extract a measure of uncertainty from these models using a Bayesian approximation of Gaussian processes. And we are able to use some state-of-the-art models to visualize what is the feature importance of our input. That is to be able to interpret on what information, visualize on what information this prediction is being done. And as soft statements that I can make is that indeed the uncertainty, predictions with high uncertainty give you a good indication of the quality of the prediction. So false positives are sometimes are usually come with high uncertainty values as well. That indeed these models, they exploit the edge and texture and shape of the objects that we need to detect to make a predictions, which is encouraging. And as I mentioned, this is an analysis that is ongoing for a couple of data sets, and ideally we would like to make this analysis public through a reproducible NF core pipeline. And with that, I would like to thank you, and I think that is it from my side.
Yeah, thanks a lot, Luis. Really interesting talk and really interesting topic. So I think it's really excited to see what will come there in the future. I think we have time for one question. Andre asked, how low in sampling can you go to train your models? What is the minimum number for a still great prediction?
I'm sorry, again, could you repeat the...
Yeah, sure. You can also check out the chat, but I'll just repeat again.
Okay.
The question was, wait a second, no, my chat disappeared. So basically the question was how many samples you need for good predictions.
That is a good question. Usually for these data sets, we're quite restrained in the number of samples, certainly in the amount of biological samples. So the the pathology data set, the tissue H&E staining data set, that data set is around 30 images, so 30 biological samples, so it's quite small. However, the objects that need to be detected are relatively small, so you have a lot of nuclei in one image. So there's mainly what happens is that we get to these high performing measures by doing dataset augmentation, but it really depends on the task and the dataset. However, I would say that sometimes with making reasonable assumptions and proper augmentation techniques, you do not need an extremely huge data set. I mean, again, depends on the task. For the leads data sets, for the computer tomography data set, those are 130 tomograms, so it's also relatively small. Yeah, it's hard to say. Hopefully this was useful.
Yeah, I think it was quite useful, at least for me. Yeah, thanks a lot again. And I think that was our last talk. And with that, our workshop is not over, but I'll still hand over to Sven Nansen for the wrap up. And remember that there will be breakout sessions in the end, so you can get the links on the website, but they will also be posted here in the Zoom chat in a second. And yeah, with that, I just hand over to Sven